Hi, listeners. It's hard to believe, but New Frontiers in Functional Medicine has been going strong since 2015. With over 200 episodes and over 2 million listens, we are growing every year. My goal is to continue to bring you the best minds in functional medicine, epigenetics, and longevity. I want the content to be actionable for the functional medicine clinician and interesting for the rest of us. Thank you for supporting this podcast with your likes, with your honest reviews, with sharing the episodes with your friends and family. And if you haven't done so, please consider leaving a like or leaving us an honest review wherever you've accessed this podcast. Thank you, thank you, thank you to our sponsors. Because of our sponsors, these are companies that I trust, that we vet carefully, that we use in clinical practice, that I use personally. Because of them, everything associated with this podcast is free. If you haven't done so, I suggest you head over to DrKF to the podcast page where you can access the full transcript for every single one of these 200 episodes. And along with that transcript, you'll get an episode summary. You can get the research citations and additional content that is pertinent to the episode. I myself return to the transcript page all the time to pull out a little snippet of information that I want to recall or access a citation. Or better yet, sign up for our newsletter, again, at DrKF and every single podcast episode will drop in your email as soon as it's released. Thanks again. Thank you to our Diamond Level sponsors, Biotics Research, Rupa Health, and Integrative Therapeutics. For over 40 years, the foundation of biotics research has been innovation and quality. Their goals remain unchanged: innovative ideas, carefully researched concepts, and product development with advanced analytical and manufacturing techniques. Biotics nutritional products are of superior quality and effectiveness. They're available exclusively to healthcare providers. Find them at bioticsresearch.com. I also want to give a shout out to our friends over at Rupa Health. They make lab testing easy, fabulous, doable for you, the clinician, and for you, the patient. Consider using Rupa as your super, super, super smart solution to all things laboratory testing. Visit them at rupahealth.com. For over 35 years, patients and clinicians have trusted Integrative Therapeutics for their dedication to quality, potency, and accuracy, and for bringing us exclusive products backed by science. From sourcing of premium raw materials to their meticulous manufacturing processes, they prioritize consistency, efficacy, and safety because our patients deserve nothing less. Find them at integrativepro.com. That's integrativepro.com. Thanks to our gold sponsors, Timeline and OneSkin. Timeline, fueling cellular energy for a lifetime of healthy aging. Find them at timelinenutrition.com. And OneSkin, skin age reversal technology. Find them at oneskin.co. Hi, everybody. Welcome to New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And today is no exception. If you're with me on YouTube, you can see that I am, sitting next to remotely the extraordinary Dr. Matt Kaeberlein. I'm excited to, for any functional medicine people out there who haven't encountered his work yet, I'm really thrilled to bring him to our community. Let me give you his background. We're going to jump right in because we've got a whole lot to cover today. Dr. Kaeberlein is the Chief Executive Officer at OptiSpan, Affiliate Professor of Oral Health Sciences at the University of Washington. He's the co-founder of Dog Aging Project and Dog Aging Institute. Dr. Kaeberlein's research seeks to define biological mechanisms of aging and to facilitate translational approaches that promote health span and improve quality of life for people and for companion animals. I want to go on and say that he is a fellow of the American Association for the Advancements of Science, the American Aging Association, and the Gerontological Society of America. He's published more than 250 papers in the peer-reviewed literature on longevity, and he's received a zillion different awards, which if you want to read through it, it's really impressive. It's over on our show notes. I've been following your work, Matt, for a long time now, and I just appreciate you. As I just said offline, your I don't know, your voice of reason. And I think more and more, we need kind of rational sort of, it seems like you take a breath perhaps before you respond, but just kind of like, I look to you to really kind of guide where we're going. And it's funny, because I didn't actually anticipate starting with this, but in this guidance, you're part of the Biomarkers of Aging Consortium. You're right in the middle of getting the language down, the biomarkers down for what aging is. And you spent your entire career really studying the mechanisms of aging, and I want to talk about all of it. So let's start by what you think aging is, and then I would just kind of like to jump into how we're thinking about measuring it and languaging it and all of that.
Sure. Yeah. Well, first of all, thank you for that kind introduction. And I do try my best to be precise in language, and I think that's one of the things that, especially in a field like aging or longevity, becomes really important, because definitions matter. And I think in this space, not everybody has the same definitions. So I'm glad you actually started with what is aging. So when I think about aging, my training, since I was a graduate student, really has been focused on the biology of aging. So I immediately approach it through the lens of biological mechanisms. And again, my training is in biochemistry and molecular biology and genetics. So that's the way I think about aging. What are the biological processes that contribute to all of the functional changes and, in general, functional declines that go along with the aging process? And again, just to kind of start from foundation, it's useful to differentiate between chronological age and biological age. So I'm immediately gravitating towards biological age because that's what I think about. Chronological age, of course, is what most people think about, which is just the number of birthdays you've had, the length of time that you've been alive. What's important to appreciate is those two things don't necessarily have to be equal. In other words, some individuals will age biologically faster or slower than the population average. So your chronological age can be different than your biological age. Some animals will age faster or slower than other animals. I always like to point to dogs, both because I'm a dog person and I've studied aging in dogs, but also because everybody gets this idea that, one human year is about 7 dog years. That's just all we're saying is dogs age biologically about 7 times faster than people do. So I think it's important to differentiate between what we're talking about. So when I talk about biological aging, I'm talking about the biological, molecular, biochemical processes that lead to the functional declines that go along with aging, and in turn lead to exponentially increasing risk for essentially all of the major causes of death and disability in developed countries: cancer, heart disease, dementia, kidney disease. You can go down the list. It turns out 9 of the top 10 killers in the United States have this relationship, that they are largely driven by biological aging. So I think where you were going when you were talking about the Biomarkers Consortium is, how do we measure that? And that's kind of one of the big outstanding questions in the field these days, is what are the right biomarkers that we can use to actually measure biological aging? And we could get into a deep discussion about this. I think I'll just stop here by saying that many people tend to gravitate towards molecular markers. That's where the epigenetic tests come in. But you can use all sorts of different biomarkers of aging. It turns out AI, but just based on facial images, can do a pretty good job of estimating biological age. So there's nothing special about, I shouldn't say that, I should be careful. There's nothing intrinsically special about epigenetic molecular marks versus metabolomic molecular marks versus functional measures versus imaging measures of skin age. They all are different, I believe, reflections of biological aging. And I would say, I don't think we as a field have a consensus definition for how to measure it, or even necessarily exactly what biological aging is. I think we're still figuring it out.
So, yeah, all right. And so then we can talk about what actually reversing it is or slowing it down. I mean, if we don't have it measured, if we don't have it defined yet. But before we do, I just wanted to throw out there too, that 2016 paper that I'm sure you're familiar with, which identified that, we can visually see people who, we can look and see folks who are aging at a faster rate. I mean, it's like we're evolutionarily or biologically hardwired to kind of see folks who might be weaker, which I think it's kind of cool. I mean, it's logical, but it's also sort of extraordinary.
Yeah. I think we have to be I think we have to be a little bit careful though to recognize that we can do that as human beings, but there are sort of error bars on that. The precision is not that great, or accuracy, I guess I should say, is not that great. And AI can actually do better than the human eye can in terms of accurately predicting chronological age. So this is where I think the question of, can we actually predict biological age, and what do we mean? And I think the field right now is struggling with this question of, are we actually measuring biological age, or are we measuring health? Some sort of health status, some metric of health status. And are those equivalent? And they don't necessarily have to be. Again, health generally declines with chronological age. It generally declines with biological age. But is health status equivalent to biological age at either the whole organism or the individual organ level? That's another level of complexity. Sorry, I keep going off on these tangents.
It's really interesting though. So
it's super important to appreciate that. I think we're starting to understand that at the molecular biochemical levels, not every organ or tissue in the body is aging at the same rate. And so your health status can be largely determined by whatever your rate limiting organ is, right? Or at least your risk of dying. So health is not necessarily equivalent to biological age. And we'll get into reversing aging. Is that even a valid thing to say? But I think the way I think about it is, if we just leave biological aging out of the equation, nobody would be surprised that you can take an unhealthy person and they can get healthier, right? Like, that's obvious. You take a sedentary 65-year-old who's eating a crappy diet, you get them to start eating right, sleeping well, exercising regularly, they're going to get healthier. Did their biological age reverse? I don't know, but they got healthier. And I think we sometimes, get those things mixed up and start claiming that we've reversed biological age when all we've really done is seen somebody get healthier.
But why don't you know that has reversed bio age? I mean, why don't you
because we don't know how to measure biological age.
Okay.
It's a chicken and egg sort of thing.
Yeah.
But if there's no consensus on how
they're likely going to live longer, right? If you've turned the volume down.
If they've gotten healthier, but that's because health status generally declines with age. Again, we didn't reverse chronological age. So there's a correlation between health status and chronological age. Nobody's going to argue that we've made time go backwards. So have we actually made biological age go backwards or not? I think is an open question. And I don't know the answer. I think that's fair to say. What bothers me is people who claim that they do know the answer and claim that they have reversed biological aging.
Well, but you don't know the answer because we don't have the tools to measure it. So it's not that it's not happening. It's just that you're not keen on the current toolbox. Would you say that's true?
I think that's fair. And I also, but there's two other things I would say. One is, I think if you're going to make extraordinary claims, you really should have extraordinary evidence to support it. Claiming that you have caused somebody to age in reverse, to me, seems pretty extraordinary, and they don't have the data to support it, I would say. That's number one. Number two, so far, we have yet to see anybody, no matter how healthy they are, that makes it to 85, 90 years old without significant functional declines. If you were really reversing biological aging, and in theory could keep doing that, you should in fact look younger in every way, function younger in every way, and in theory be able to maintain that. I'm not saying it's impossible. There's nothing biological that says that's impossible. It hasn't been done yet, though.
What, oh my God, you're just like making more questions for me here. Um, okay, so I want to ask you about the blue zones, but just put a pin in that.
Sure.
Because now with what you've said circles me right back to the biomarker consortium. And what are you guys actually talking about? What's going on over there?
Yeah. So I think the way I would describe this is a collection of mostly academics and scientists who are all trying to grapple with the challenge of developing something closer to a consensus for first of all, what we mean by biological age. What are the tools that we have available now? And what questions do we need to answer before, as a field, we can feel confident that not only are we able to measure biological age in some meaningful way, but start to move towards where those tools can actually be used in either and a clinical practice in a meaningful way, in an actionable way, and also for regulatory bodies to hopefully give us ways that we can assess whether or not interventions are actually impacting the biological aging process. And so, the obvious case use would be a company that has a drug that they think modulates the biological aging process. They would like to get it approved by FDA to be used clinically. Right now, there is no path for that company to get their drug approved beyond picking a existing disease or functional endpoint and doing a typical clinical trial. So is there a future where something like the epigenetic clocks or some collection of different biological age metrics could be used in a clinical trial where FDA would accept those as surrogate endpoints, meaning that there's a high level of confidence that this is expected to actually improve health, reduce mortality, improve quality of life? We're not there yet. Sees that as a goal that is possible, and starting the discussion now makes a lot of sense to get to a point where the regulatory bodies will have confidence in these markers.
That's awesome. That's awesome. And it's a really diverse group of academics. I would imagine you guys have some pretty interesting conversations over there, some really interesting dialogue going on. But you're defining what the job is ahead, and then you'll begin to pool together the relevant biomarkers. Cool. Yeah,
Yeah,
we'll stay tuned.
I also would say, I mean, these kinds of things, you're never going to get 100% agreement, but I think
Right.
the conversations and the documentation that can come out of those conversations are valuable in and of themselves, and
I
also
want to,
pointing out where there is disagreement.
for sure. Yeah, yeah. And I mean, I just want to say like the sort of the obvious for anybody who's familiar with my publications. Of course, we talked about potential reversal of biological age, which um, really put the stone in some people's shoe. Uh, 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 and I just want to acknowledge that up front. I am, however, I'm a clinician
Yeah,
and was following the voice of the literature at the time. But I
sure. And I mean,
appreciate it. I
and
appreciate the conversation.
I would say again, um, everybody's entitled to their opinions. I'm just sharing mine, but I think,
Yeah. Mhm.
I tend to have had similar sorts of conversations around the word health span, and I've been guilty of it. Like if you look at some of my papers in 2016 and before, in the title, we said we improved health span. And then I realized there is no consensus for how we actually measure health span. It is a useful
Right.
concept. Everybody
Right.
kind of gets what we mean. There is no agreement at all in the scientific community for how you measure health span. As scientists, we should not claim we have statistically changed something that we can't actually measure, right? So I think we all know what we mean when we say we've improved health span, but we should not claim that we have shown a drug or an intervention improves health span in the scientific literature if we don't have a consensus way to measure it. And I kind of feel the same way about biological aging. It's perfectly appropriate to say we have reversed this metric of epigenetic age,
Yes.
right?
Yes.
Because we
Yes.
all know exactly what we're talking about,
Right.
but we don't actually know that epigenetic age, by whatever epigenetic test you used or collection of tests, is truly reflecting biological age of the entire individual or animal at this point. And so we just have to be careful about the the way that we portray ourselves, particularly, I think, in the scientific literature.
And I think that's absolutely a fair criticism. And it's really nice for us to spend some time here teasing it out, because I think, on this podcast, there's a lot of clinicians listening who are probably aware that there's a challenge around language, but not really the detail behind it. So your extremely clear explanation is useful. So I guess, so I have a couple of questions on that. Arguably, blue zoners are living longer. They are clearly living longer by definition, and they have a better health span. I mean, there's certain characteristics. Now, why is this not something that you can dive into? I mean, speak to blue zone.
Well, I think there's a couple things to say. So I think the blue zones themselves have different levels of confidence and credibility, right? So there have been questions raised about some of the blue zones.
That's right.
So, I mean, the one that I feel probably, and this is, again, my own opinion, and mostly based on just what I know about Okinawans, that one to me, I think, is kind of the gold standard, where there's really no question, at least historically, Okinawan Japanese lived significantly longer than mainland Japanese, who already have a pretty long life expectancy by international standards, right? Yeah. And so there, I think you can clearly say there's something about that population that was different. And again, to the extent that we have quantitative data on health span metrics, they seem to enjoy better health later into life as well. So what are the likely explanations? The most likely explanation is caloric restriction. And that's probably not the entire answer, but it is the case that Okinawa, again, up until about probably 30 or 40 years ago, at least, is sort of one of the largest pure caloric restriction without malnutrition natural experiments to have ever existed. They ate, I don't remember exactly how much, but it's on the order of 15-ish% fewer calories than mainland Japanese. Very healthy diet, lots of seafood, lots of complex carbs if they're eating carbs. But they also exercise, not in a like the American exercise weekend warrior or, go to the gym, but just as part of their natural daily lives, lots of walking around. Also had strong social connections, right? So again, it tends to come back to what I think most people would accept as the commonalities around what does a healthy lifestyle mean. It means lots of natural food, lots of vegetables. If you're eating a lot of meat, typically richer in seafood than in red meat, although we could talk about red meat. I think red meat's an interesting topic as well. Regular exercise, strong social connections, right? Relatively low stress lifestyle. Those seem to be the commonalities that come out of a lot of the blue zones. And I think you can point to Okinawa. But again, what makes Okinawa interesting is they also ate less. So, and there could be a genetic component as well. So those seem to be the commonalities, in my view, to a lot of what people attribute to the blue zones. And I think that fits pretty well with what most people would agree in general are the themes of a healthy lifestyle, which is likely to benefit people in the United States if we could get people to actually do that.
Right. Now, what about diving into the UK Biobank data and seeing if you can tease out health span from that massive, super interesting data set?
Sure. And again, people are starting to do these sorts of large scale epidemiological studies. We have to be really careful for obvious reasons with those types of correlative studies. There are all sorts of ways that the analysis can go wrong, right? Including who are you comparing to? I think here's an interesting example. And again, it's not just the UK Biobank, but you see a lot now alcohol consumption. Okay, we're getting a whole nother topic, right? Alcohol consumption, right? So the question of, is moderate consumption of alcohol good for you or not, right? And again, there was a study not too long ago from the UK Biobank that addressed this, which is what made it pop into my head. But when you're thinking about challenges with the way you do these studies, the consensus now seems to be, I don't know if it's consensus, the popular opinion seems to be moving towards no alcohol is optimal, right?
Yeah.
The problem is the data hasn't really changed. The problem is the group you're comparing to. So the data is pretty clear. A low moderate, depending on how you define moderate level of alcohol, gives you the lowest all-cause mortality, the least likelihood of dying, right? When you compare to people who don't drink anything, it turns out that moderate consumption of alcohol is beneficial, right? If you move what you're calling zero to something that's not zero, then it's no longer beneficial, but it's because you've changed the comparison group, not because the data itself has changed. So the interpretation of those kinds of studies can be very, very dependent on the population you're comparing to. I think this is where I was going with red meat. I think that's another example where, if you compare people who eat a lot of red meat and also eat crap for the rest of their diet, red meat's bad. Yeah. Protein's bad. If you compare people who eat a lot of protein and eat crap and are overweight, high protein's bad.
That's right.
If you compare it to people who are eating a healthy diet, those trends tend to go away and it becomes unclear, right? So I think we just have to be really careful with the way we do those sorts of correlative observational studies. So yeah, there is a lot that can be done, but I feel like it gets misinterpreted more often than it gets interpreted properly. And so honestly, sometimes I just throw my hands up when I see this stuff show up on CNN. I'm like, another one.
Right.
So sorry, that didn't really give a lot of clarity.
That's fair.
But UK Biobank's interesting for a variety of reasons, because you can do all sorts of analyses, all of which have these risks associated with them. But I think when done properly, you can start to tease out some interesting signals around. I don't know if you saw this study where they looked at prescription medications, right? And basically looked at all of the prescription medications that have been prescribed. I think more than, I don't know if it was 1,000 people or something like that, some lower number, and tried to compare people who got those prescriptions to health matched individuals and ask, was there any relationship between the medication itself and all-cause mortality or risk of dying? Most medications, mortality went up, which is kind of what you'd expect, right? But for a few, mortality actually went down. And it's a really interesting list because several of them overlapped with things that have been shown to increase lifespan in mice, like SGLT2 inhibitors, like estrogens. It's suggestive that maybe there was a biological aging signal there, because completely independently, those things in laboratory animal studies had been shown to increase lifespan. So I do think there are some really interesting, cool things that can be done with UK Biobank is a really great example where the data has been made available to the research community. And so it's very rich and there are opportunities there.
Yeah, there's studies coming out all the time. And to your point, they're not always really useful, like the fish oil AFib, but that is,
I think you have to be cautious. Yeah. And this is one of the challenges in this space is, I struggle sometimes with knowing what to believe. How does somebody who can't dive into the literature, doesn't have that background, doesn't have a medical background, how do they know where they can get, really believable information? Like, who are the credible voices? And even the credible voices get it wrong sometimes. So it's a real challenge. I guess my advice would be don't take anything off of one study for sure, right? No matter how strong it seems, especially if it's a purely epidemiological study, don't take that as the gold standard if it's one study that, that everybody is pointing to. Really, we need to see this be reproducible optimally across populations, or at least line up with other lines of evidence that are all pointing in the same direction.
But yeah, I think that's fair. It leads me to another question, but I'm going to digress. I'm going to bring us back to where we were, because I want to ask you, you're talking about, defining health span and defining biological aging. I mean, and actually this kind of, abuts what you just talked about. I mean, science is medicine in general. We're at this extraordinary inflection point, where we're able to measure and see stuff, and God, it's exciting. It's incredible. We're starting to layer AI on top of this. And, and so it's definitely more of a moving target now. And so grappling with definitions and really kind of keeping your eyes open and, and moving intentionally, I think is important, but it is really, very, very exciting. But I can see why, we need the consortium. Like, we need folks like you really kind of cutting the ground for us. But to that point, so defining biological age, defining health span, I think, yeah, it seemed to be not sort of a safe thing to talk about aging itself, biological aging, as being a program phenomena, as there being like sort of two tracks, right? So there's aging, like I think humans can max out at about 120, whereas Greenland sharks can go for 450 years. Like or naked mole rats are going to live to be like 30, which is nuts. But there's just all sorts or a fly, right? A fruit fly doesn't hang around very long. There's these very different, very sort of reliable lifespans that we have, and there's reason to think that it's programmed, that it's really hardwired, and this would make defining identifying, measuring biological age difficult, because then we have exposomic aging, which is all of the environmental inputs. So you take somebody who's hardwired to age to year approximately year X, and then but they're smoking, et cetera, et cetera. So there's like these two trajectories that I think add to it, and I want to get your thoughts on that.
Sure. So I think the first thing to say is most of the discussion around programmed aging in the scientific community, again, really comes back to definitions. When people say programmed aging, I think where the disagreement arises is technically that means that natural selection acted to cause a certain rate of aging. I shouldn't say anybody. I don't think most people who understand the biology of aging believe that's true. By and large, natural selection is acting on the factors that will influence whether or not you reproduce at the right time and in the right number optimally, because then you can outcompete your competition. It doesn't have anything to do with how long you live. There may be rare cases, but by and large, evolution, natural selection is not controlling the rate of aging. That doesn't mean that aging is not genetically controlled. It just means those genes weren't selected for that. They still control aging. And that's why we see this coupling of development with longevity, things like mTOR, insulin signaling, IGF-1 that affect
factors.
Both. Yeah, right. So they were selected for something else, but they do control aging. So in that sense, if that's what you mean by there's a program, yeah, aging is under genetic control to some extent. Longevity is under genetic control to some extent. And I think when we look across species, like anything else, the differences in maximal life expectancy across species, which is what you were referring to, a fly, a dog, a person, a clam that lives 500 years, that's going to be a whole bunch of genes. It's not a single gene that determines those differences, which I think is why we haven't been able to recapitulate that level of lifespan difference in the laboratory, where we can manipulate one gene or a few genes at a time. And it's not going to be primarily environment that's controlling those species level differences. So I think one of the interesting questions is, within species, I think we've got a pretty good handle on kind of what the fundamental network of genes and proteins are that modulate the rate of aging, that determines the difference between a person who's going to age and die at 65 versus age and die at 105, right? We understand that in mice, we understand that in flies, we understand that in worm. With some, I mean, obviously there's a lot we don't understand, but we can put names on it. We know a lot of the players. We know some of the genes to tweak to change lifespan by 25, 30, up to about 50% in laboratory animals. What we don't understand is the biological differences that go across species by an order of magnitude or two orders of magnitude. And the question, one really interesting question is, are those fundamentally different mechanisms than what controls within species rate of aging? I think they are. I think we have no understanding of what those mechanisms are right now. What's also interesting, though, is that what seems to control within species rate of aging seems to be highly shared, from nematode worms to fruit flies to mice to dogs and probably people. Insulin signaling, mTOR, IGF-1. Like, we have data across all of those species. Those things regulate the rate of aging of differences in rate of aging of individuals within the same species. So I think that seems pretty clear. We haven't proven it in people, but there's enough arrows. We were talking about if you get multiple arrows all pointing in the same direction, there's enough arrows pointing in the same direction that I think in people, it's probably true that the same pathways that modulate aging in mice and worms and flies to some extent also modulate human aging. But one of the implications is we're probably not going to do better than 20% at best in people by tweaking that network. So that's about what we can do pretty easily in mice. So we're probably not talking about doubling lifespan, tenfold changes in lifespan, because we just don't understand those mechanisms. You mentioned Yamanaka factors. We can talk more about that. That's the one area that I think people are starting to have thought may give us much larger effects on health and longevity. I don't know, but I think the early data, and it's still early, is suggesting we may end up bumping up against that same wall. We may end up bumping up against that 25% effect on lifespan, even with the repeated partial epigenetic reprogramming. So we'll see what happens.
What suggests that wall to you? What is it that's creating that opinion?
So there was a panel discussion recently, I think it was at the Aspen Institute event, where Rick Klausner from Altos is a large company that's doing some of the leading work in the epigenetic reprogramming area, where he actually said they are able to partially epigenetically reprogram mice in the laboratory multiple times. The mice seem to have greatly improved health span, but they're not doing any better than 25% effect on lifespan. Now, it's all unpublished, haven't seen the data, so take it for what it's worth. But that fits with what little is in the literature with partial epigenetic reprogramming, which is not as good as rapamycin even. So far, nobody's been able to get past that barrier. The only thing really that's gotten past that 25% mark in mice, and again, I'm being sloppy in my terminology because percent is sort of not the best way to look at these numbers, because percent is affected by the denominator, which is the control lifespan. But for sake of simplicity, let's say 25%. The only thing that really does better than rapamycin is extreme caloric restriction in mice. Nobody has done better in mice than an experiment, to the best of my knowledge at least, that was done in the 1990s by Rick Weindrich and Roy Walford, where they restricted mice by 65%, and they got about a 65% increase in lifespan. Nobody's done better than that in the last, what are we going on, 30 plus years now. Again, I think we should ask the question why. And one interpretation would be we've kind of bumped up against the best we're going to do by tweaking this same network. We really need to understand what else is acting at the level of across species if we want to do better than that upper limit. Again, that's speculation on my part, but so far all the data supports that.
That's interesting. That's really interesting. So we're not hitting escape velocity and
there is no data to support the idea that longevity escape velocity is getting closer. Again, if you want to believe something in the absence of data, that's called faith. That's fine. I have nothing against faith, but it's not science.
I had a really fun podcast with Vittorio Sebastiano, who you're in the consortium with. You're familiar with his work, and one of the, I thought that his comments around focusing on specific organ rejuvenation using Yamanaka interventions was compelling. Like in women, ovarian rejuvenation just strikes me as extraordinary. So if we can keep our ovaries younger for a little while longer, we will influence brain, we will influence bone, we will influence heart, not to mention the obvious reproduction as well, if we choose to go in that direction. Thoughts on that? And that sort of goes into the health span conversation, which we're still defining, but thoughts on that?
And they're tied together. I mean, again, I agree completely. I think that, and I don't, when I talk about the partial epigenetic reprogramming, and I'm saying I don't think we're going to double lifespan, that's not at all meant to suggest that it's not a hugely potentially powerful therapeutic approach. I think there are lots of potential applications. We'll see. There are also some challenges, but I'm very enthusiastic about the idea that that these approaches can have significant effects on a whole bunch of health conditions and improving health span. It turns out there's a whole body of literature, certainly starting with the heterochronic parabiosis, which is the studies where you take an old circulatory system and hook it up with a young circulatory system, and you can get positive benefits on the old animal and negative effects on the young animal. So there are circulating factors that can influence whole animal health with age, right? Again, you bump up against the same limit in terms of effect on lifespan with heterochronic parabiosis. You can also do that by taking young ovaries and putting them in an old mouse. Again, you bump up against the same sort of limit. So yeah, you can have effects on health span and lifespan at the individual organ level, and also potentially at the whole body level by rejuvenating certain tissues or organs. I have seen nothing that makes me think that approach is going to get us past the same sorts of limits that you bump up against so far, at least in mice with rapamycin or caloric restriction or reducing IGF-1 signaling. But yeah, I think there are lots of opportunities from a therapeutic perspective, especially as we're starting to learn about differences in aging rate in different organs in different people. Like, it may be that in me, it's my kidneys that are aging more rapidly. And so if we could have a therapy that could go in and take care of that, that would be fantastic, right? And could significantly increase my individual longevity and health span. So I think there's lots of applications there for sure.
It's really exciting. To your point, I mean, it's almost more satisfying to look in that level versus, can I tack on another 30 years? It just makes more sense.
I tend to, again, it's kind of funny because I sometimes get branded by the extreme life extension people as being a pessimist, but I'm not at all. Like, I think there are huge opportunities to have large impacts on health span and potentially longevity for a lot of people. It's just, I think 20 years of extra health is a big deal, right? And I don't think we should minimize it because there's no evidence that we're going to get 200 years of extra health. So I think we have to set our expectations in a realistic way and not only not mislead people, but not turn people off by talking about stuff that sounds more like science fiction and in many ways is science fiction when the science fact is actually pretty darn exciting right now.
It is. It's extraordinarily exciting. And quality of life in the US as we age really sucks. It's not a fun place to be by and large.
So the statistics are pretty daunting. So I was actually pretty surprised, when I looked into this. So the CDC says that 60% of Americans suffer from at least one chronic disease.
Yeah.
The median lifespan or median age in the United States is 38 point something, right? So if 60% of the people have at least one chronic disease and half the people are younger than 38, and the average life expectancy is mid-70s, that means that most people are living 3 to 4 decades with at least one chronic disease. So I sort of think of health span as harder to define, but if we define sick span as the period of life with at least one chronic disease, that's a pretty big chunk of life, right? Wow. That I think a lot of people don't realize the impact that has on their, not just the quantity of their life, but the quality of their life. And much of that time people can reclaim. Like, we have the tools now to get that back for most people. That's where we should be focusing our attention, in my view.
Healthcare providers have long relied on Dutch to uncover the underlying causes of hormone-related issues, offering actionable results supported by peer-reviewed research. In research highlighted in the November 2023 edition of the journal Menopause, dried urine testing was validated as an effective method for monitoring hormone replacement therapy. This pivotal study is just one example of the rigorous research that forms the foundation of Dutch's approach, empowering providers to make informed decisions with certainty. This study underscores Dutch's commitment to evidence-based medicine, equipping providers with the confidence to navigate complex treatment decisions. Explore the world of Dutch testing and our steadfast dedication to scientific integrity at dutchtest.com/research.
Dr. Fitzgerald deeply researched how to age well, and a clinically studied formula that can do wonders for your aging process is Qualia Senolytic. Senescent cells, or zombie cells, accumulate in your body past age 30, wasting your energy and nutrition and contributing to accelerated aging. Qualia Senolytic combines 9 vegan, plant-derived compounds to help naturally eliminate senescent cells to help you feel years younger. Try Qualia Senolytic at qualialife.com/Cara15 with code Cara15 for 15% off. That's Qualia Senolytic at www.qualialife.com/Cara15.
You need to make a slide for that. Have you noticed that there's been a wholesale adaptation of your bio-age
Yeah.
and chronic
Yeah, it's
like
nice to
been...
see. It is the first time I ever, this was, 15 years ago now. The first time I ever saw one of my slides show up in somebody else's talk, I was like, what the hell? They stole my slide. And then I was like, okay, that's actually the best form of flattery. So I think that's great. Yeah.
Yeah. I'm just working on a deck for a nutrition group in the UK, and your slide is in there.
Yeah. Everybody, you're free to use my slides. Okay, go for it.
But create that one. It's incredibly powerful.
Yeah.
And then one diagnosis begets another. You really slide down a slope, I think.
Right. I think so too. Yeah, absolutely. And again, it's maybe not surprising that one of the first chronic diseases that a lot of people experience in the United States is metabolic related, obesity or diabetes. And I think there's pretty good reason. Again, this is a little bit of speculation going back to my sort of insistence that we don't overinterpret how we define biological age. Little bit of speculation. I think there's reason to believe that obesity and particularly metabolic disease accelerate biological aging. So it's not shocking if that's the first thing that people experience, that's going to increase the rate of onset of comorbidity of other age-related diseases. And that's also, I think, why, you're starting to see headlines around GLP-1 agonists maybe reversing aging. I don't think they're reversing aging. I think they're reversing an acceleration of aging, right? They're slowing down an acceleration of aging. It's an interesting hypothetical. In normal weight people, would GLP-1 agonists reduce mortality and reduce age-related diseases through a caloric restriction effect? I don't know the answer to that. But I do think in people who have metabolic disease, for sure, you're going to have a positive impact on a bunch of age-related diseases if you can reverse that metabolic disease. There are other potentially negatives to the GLP-1 agonists, but they're super powerful drugs from that perspective.
Super powerful. And it's another conversation that we can talk about. I think primarily it would be, muscle and so forth.
But, muscle and bone would be the two places I would be most concerned about. Hormones too, but
quick, before we talk about supplements, rapamycin, metformin, and, sort of the what to do in your opinion, I just wanted to ask you about the pan-mammalian clock, the Horvath pan-mammalian clock, just kind of linking together that some sort of shared aging phenomena that they're starting to tease out, and get your thoughts on that.
Right. So this is the idea that you can measure changes in the epigenome. And again, maybe it's worth just at a high level sort of defining what we're talking about. So epigenetics is on top of the DNA, and really to some extent can just be thought of as kind of the control knobs on individual genes. We can modify the epigenome to turn genes up or turn genes down. And there are several ways to do that. Methylation is the one that most people measure when they're measuring these epigenetic clocks. And we have tens of thousands of methylation marks in all of our genomes. Yeah. And so what has been seen is that you can find a subset of those, usually in the order of a few dozen to maybe a couple hundred of these tens of thousands, that change in a predictable way with age. And then you can go on and find a different subset that change in a predictable way with health status or with remaining life expectancy if you have biobank samples, things like that. So what has started to emerge, we talked about, first of all, you can do that at the organ or tissue level, which I think is super interesting within individuals, different signatures for different organs and tissues. You can also look across species and find a different subset of marks that seem to correlate with aging. If you sort of take what we know about the maximum lifespan and sort of normalize that all, so we're talking about the same sort of relative axis. In other words, you would, in an animal that lives 100 years compared to an animal that lives 1 year, you would kind of shrink it by 100, right? So if you do that, you can find a subset of epigenetic marks that seem to mark the aging process across mammals, right? And so this suggests one interpretation would be, and this runs against what I was sort of suggesting before, that there is a conserved biological aging process across at least all And that might be true. The challenge is there's no mechanistic connection at this point. In other words, nobody has gotten to the point where we can actually look at the specific epigenetic marks that are correlated in any of these clocks and say mechanistically, this is causing a change in the rate of aging, or a change in the risk of dying, or a change in the risk of a specific disease. So they are purely correlative at this point. And I think in the absence of a mechanistic understanding, we don't really know what it means. And one of the, I think, and again, this is, so I have a little bit of actually I have more of a mathematical background than most people. I do have an undergraduate degree in mathematics, but I haven't used it in decades. But my mathematical background leads me to recognize that the dimensionality on the epigenome is so large, in the tens of thousands, that you are very likely to be able to find a subset of marks in the order of a few dozen that will correlate with anything. Yeah. And so again, in the absence of a mechanistic understanding of whether that correlation is causal or responsive or just a correlation, it's hard to have a ton of confidence to know to know how useful that is as both as a measurement tool and as a understanding the biological process. So yeah. I think it's an open question. And sort of the other thing that maybe is worth saying, and again, Steve Horvath may have a very good answer to this. I haven't asked him this question, but one of the things that makes me a little cautious about the way the epigenetic clocks are being used and marketed today is these clocks, in order to make a prediction about mortality 10 years, 20 years down the road, in the absence of doing a 20-year experiment, the only way you can do that is by going back to biobanked samples and making predictions based on looking at samples from 20 years ago or 30 years ago. Then you can know which people died and which people didn't and come up with these correlations. My concern there is that the environment 30 years ago is so fundamentally different than it is today. It may be a completely different set of marks that are going to correlate today with health outcomes than they did 30 years ago. And so I'm a little bit worried that because we don't have a mechanistic understanding, we may be misinterpreting what these clocks are actually telling us. And they may not be telling us what many people believe they are telling us.
I think that's fair. I think that's definitely a fair pushback. I mean, the omic age clock that True Diagnostic developed with, I guess with Harvard, is using currently banked specimen. I mean, it might be onto something. It's sort of a sidebar. But I also want to throw out the polycomb recessive complex clock, looking at the entire methylome, that Sebastiano and Horvath developed, that seems to possibly be touching on a little bit more rather than, isolated set of markers correlated with the aging Germany. Yeah.
And I mean, I think this is both one of the exciting things about the field and one of the challenges is there are so many flavors of these clocks now.
Yeah.
And nobody really, at least that I've seen, has really rigorously shown how well these clocks match up to each other. So do they all give the same answers? And if they don't, what does that mean? When they don't, what does that mean? Right. Does that mean
and
the
I don't
old
think
clocks
they
were
I don't
wrong?
think that they necessarily I don't think they do, actually.
Well, okay, if they don't, then that should tell us something, right?
Yeah, what does it mean? Right. That aging is complex.
Yes, and at least some of the clocks were not being interpreted properly or are not being interpreted properly. So I think we just have to approach this with a fair degree of caution. And, again, my sort of viewpoint right now is these are really great research tools. I think they have the opportunity to be useful from a regulatory perspective in clinical trials. We're not there yet. And if we want to use them in the context of a healthcare practice, they should be used in combination with more standard, more traditional blood-based biomarkers and functional measures. The stuff that we are, I think, more confident are going to be predictors of current health status and future health outcomes, future disease risk. I would not. And if the epigenetic clocks don't match up with those other things, I'm going to put my money on those other things for now.
I think that's fair. I appreciate that. As a clinician, I'm casting a wide net of, standard biomarkers. I do think there's a place for at least some of the clocks out there. And I'm excited to, to pay attention to the others. Like for example, I would like to send our IDAT files from our original cohort to to look at that polycomb clock, looking at the whole methylome. So anyway, I'm going to put a pin in this because we're getting pretty arcane, and I want to circle back onto like some easy, like some ground that people can relate to. But it's a cool space and a cool conversation. So let's talk about interventions like rapamycin. You're really bullish on rapamycin, and more studies are coming out in humans. For a long time, it was just like, we have no idea. But now I think we're on firmer ground, and you're bullish on it. Talk to me about what's up in the scientific community with it, and why you like it.
Sure. So rapamycin, there's a few reasons why I guess I'm bullish. I mean, I still say, with complete sincerity, we don't know the extent to which rapamycin is going to increase lifespan, certainly in people, or improve health span for most people. I am 100% confident that rapamycin can improve health span for at least some people, because I'm one of those people, and it had a significant impact on my health span, which I've shared, and I'm happy to share again. So I think the question is, to what extent is rapamycin really slowing biological aging in people? And can we start to get to a point where we can have some real rational risk-reward analysis, where we can be confident which people does the reward outweigh the risk, and what is the risk actually? Why am I bullish on rapamycin? So in the preclinical studies, preclinical meaning, everything in laboratory animals, mice, flies, worms, yeast, marmosets now, rapamycin always increases lifespan, always improves multiple health span metrics. Does it improve every age-related metric? I don't think we know that, probably not. But certainly most age-related health metrics in most tissues and organs in laboratory animals seem to be improved by rapamycin treatment. You can start the treatment in middle age and still get most or all of the benefits for both lifespan and health span metrics, at least in a mouse. We now have data in a non-human primate, in a marmoset, showing lifespan extension and at least some aspects of health span being improved. We have some data in dogs, similarly evidence for improved health span. And I think we're learning that the side effect profile of rapamycin, which is what the biggest concerns were, I think, for a long time, at the doses that people are using off-label, where some people, I believe, are seeing benefits, really the side effects are pretty minimal. Not significantly different than placebo in controlled studies where there's been a placebo. The one side effect that really looks real to my eye is about a 15% likelihood of developing canker sores in people using rapamycin off-label. So I think we can start to have some confidence around safety. And there have been a few studies now in people suggestive of efficacy. So we published a study last year looking at about 300 some people who'd been using rapamycin off-label, compared them to people who'd never used rapamycin. Take it for what it's worth. There are lots of caveats with that kind of data, but it was suggestive that rapamycin had positive impacts on things like chronic pain, maybe even things like depression. I think the strongest data was around severity of COVID-19 infection and potential for long COVID. Again, that lines up with other data, biologically plausible. There have been a couple of clinical trials now, these are years old now, from Joan Manick, suggesting that a derivative of rapamycin can boost vaccine response in healthy elderly people. The Pearl trial, which again has lots of issues, the biggest probably being that the dose was lower than what most people are using off-label. Again, no real evidence for side effects, some evidence for improvements in body composition, at least in women, meaning preservation of lean mass in women taking rapamycin, or maybe even improvements. Again, that lines up with anecdotal data that many people report these positive changes in body composition when taking rapamycin. So I think again, none of it is great, right? We don't have the definitive randomized clinical trial data. Again, I should just say this, clinical trials get it wrong all the time. So I don't put as much faith in clinical trials as some people do, but it would be great if we had really large randomized clinical trial data giving us information to go off of. But the data we've got, I think, is all pointing in the same direction, that at least for a subset of people, Rapamycin, most people in the once weekly 5 or 6 milligrams a week, plus or minus a few, right? It's messy, can get some positive benefits. And my intuition is it tends to be people who have a high chronic inflammatory burden that feel better when they're taking rapamycin. And again, that lines up with the biology of what we know about rapamycin. So I think the preclinical data, which is rock solid, rapamycin is without doubt the gold standard for a longevity health span intervention in laboratory animals, combined with what's starting to emerge in the clinical world, is pretty suggestive in my view.
That's awesome. Okay, great. And you actually answered my questions on dosing and I mean, what about folks suggesting that one take a break? Is that something?
Right. Again, nobody knows. So
Yeah.
in laboratory, and that's what I do. I typically will do 3 months on and then some period of time off. Although last time I did 6 months on. So again, it's all like everybody's kind of doing their own thing. There is no right or wrong dosing. Nobody really knows. 6 milligrams once a week is the most common dose. Is that too high, too low? Should women take less than men? Like nobody really knows. But I think that dose is pretty safe for most people. Should you take a break? So what we know from the laboratory animal studies is that at least in mice, 3 months of a single 3-month treatment in middle age is enough to give a significant increase in lifespan. Is it the biggest increase in lifespan you can possibly get? Probably not. But we know that single transient treatments are enough to have a long-term effect on the biological aging process. So in people where
that? Yeah.
where we're working with an unknown risk and an unknown reward equation, you could make a case that you're going to reduce your risk of long-term side effects by doing repeated transient treatments with rapamycin. So how long do you need to get the benefits? And what I think is interesting here, and this I think comes because of the biological mechanism for how rapamycin works, which we haven't really gotten into, but for the purposes of at least right now, I think we can just say biochemically rapamycin is a very specific drug. It inhibits a protein called mTOR. mTOR does a whole bunch of stuff, but I think the biggest effects that most people experience are the ability of rapamycin quite rapidly to tamp down chronic inflammation. And particularly, it seems very good at tamping down age-related sterile inflammation for reasons that are complicated and I don't completely understand. I don't think anybody does. So it seems like, so in mice, you can get benefits as fast as 4 weeks. People have shown 4 to 6 weeks is enough to get significant improvements in some things, up to 12 weeks, or you can do continuous. In people, we don't have that data, other than from Joan Manick's studies where they used a derivative of rapamycin called everolimus, exactly the same biochemical mechanism, and they showed in 6 weeks in healthy older people that was enough to then improve their subsequent response to a flu vaccine. That perfectly matches a previous mouse study. So the time frame, even though mice are aging, let's say 30 times faster than people biologically, the time frame for efficacy of rapamycin, at least for that outcome, is the same. So you might only need 6 to 12 weeks to knock down that inflammatory burden that then is impacting all sorts of tissues and organs in humans. And again, my personal experience, so the first time I took rapamycin was after I'd been diagnosed with frozen shoulder, which is inflammation of the shoulder capsule. And that's why I took it. I was like, okay, I can't go another year with this. And the doc didn't want to do anything for me other than go back to physical therapy. So I figured I have nothing to really lose. I knew I was pretty confident that it was safe. I could give it a try. And I went in planning on 12 weeks. And within 12 weeks, I was 95% better. So I think there's reason to believe that in that 6 to 12 week time period, rapamycin at the doses we're talking about is pretty effective, at least knocking down some of that chronic sterile inflammation in people who have a high inflammatory burden. Does it have benefits in people who don't have a high inflammatory burden? I don't know.
Right, right, right. But the safety profile seems to be pretty good.
Yeah.
Like, with the exception of canker sores. I
The
mean,
one thing I'm still, I'm still on the fence about is there a slight uptick in risk of bacterial infections? Again, if you look at the data across the board, you can make a reasonable argument that there's probably an improvement in antiviral effects. So rapamycin will boost antiviral gene expression. The stuff we've seen with flu vaccine, subsequent infections with flu, coronavirus match that. But there might be a corresponding slight increase in risk of bacterial infection. It's not huge. It's not at the level of organ transplant patients who are taking daily doses of rapamycin, higher doses. It wouldn't surprise me if there is a slight uptick, though, in risk of bacterial infections.
so more information needed, more research needed, but it's cool. I appreciate your synopsis of this, and we'll be sure everybody to grab the citations and everything that Matt's referenced so far. We'll just have on the show notes. What about metformin? What are your thoughts on metformin?
So metformin is a really good diabetes drug.
Yeah.
I think the evidence that metformin is particularly good. Again, if we go back to my analogy of this network that kind of underlies the biology of aging as we understand it today, there are a few nodes in that network that seem to be very useful for tweaking the rate of biological aging. mTOR is one. AMP kinase, which is one of the targets of metformin, is also in that network. But metformin itself doesn't seem to be particularly good at modulating the network as a whole. And what I mean by that is, if you look in the animal studies, particularly mice, the data that you can significantly increase lifespan or improve health span in mice with metformin is really lacking. There's not a strong body of literature, and there's some negative data that people have been unable to accomplish that. So I think metformin is one of these cases where the enthusiasm was very high for metformin as a drug to modulate the biology of aging, generally speaking, several years ago. That enthusiasm has waned. I think that enthusiasm was in part, we talked about the challenges with human epidemiological studies, right? There was a study that was done that looked at diabetics taking metformin, diabetics not taking metformin, and then non-diabetics not taking metformin, and looked at all-cause mortality risk. And they concluded that there's no question, diabetics taking metformin versus diabetics not taking metformin, they do better, they live longer. But there was a hint from that study that the diabetics taking metformin might actually live a little bit longer than the non-diabetics. That got everybody really excited about metformin as a drug that people should be taking who are non-diabetics. Turns out that was probably wrong. So multiple studies now have tried to reproduce that and have been unable to. One, I believe, in the same population even, and then at least one other in a different population. So I think the consensus has shifted that metformin in non-diabetics probably isn't going to have a big effect on the biology of aging. And I think there's also a maybe growing appreciation that there may be some downsides to metformin that hadn't been previously appreciated. Such as potentially blunting some of the benefits of exercise. Again, you can go back and forth on that. I'm not going to make a strong case there, but I think there's some evidence to support that. Maybe some effects on hormones that are suboptimal. So my feeling is if you're diabetic and your doctor suggests metformin, it's a great drug for diabetics. If you're pre-diabetic, even, it might make sense. If you are not metabolically perturbed, I don't personally see a lot of rationale from a biology of aging perspective to take metformin.
Okay, cool. All right. I have a ton more questions, but we don't have a heck of a lot of time. I want to talk a little bit about supplements. In my world, I'm looking at the essential nutrients and conditionals. I'll do a relatively broad swath of laboratory testing to identify what the person in front of me really needs, and
Right.
and create something based on that using diet and using some supplement interventions. But there are like tons of longevity supplements, just getting, the promise of them is just so overstated and broad for everybody all of the time. And I have patients now coming to me since our publications who are focused on longevity and who are on these really classic stacks of stuff that are getting attention, like taurine, spermidine, of course, the nicotinic acid molecules, resveratrol, which sort of another conversation. But like, what do we want to be thinking about in this space? And I'm curious what you're taking.
Yeah, so I don't take a lot, actually. So I take creatine every day. I do resistance training. I think that's one where there's really very little doubt that has benefits in that context and very little downside. I take vitamin D, omega-3, to get myself into a range that I think is reasonable because I was deficient in both. I've started taking a multi. Again, I just kind of feel like that's an insurance sort of thing. Like I don't, I eat a pretty healthy diet, but again, there may be a little bit of evidence pro or con on multis, but I don't see much downside there. I don't really take supplements outside of that core with any frequency. And my rationale there is partly not because I don't believe that there's evidence to support some of these other things. I think you can find, again, this is a challenge because you can find papers that will support taking just about any supplement on the market if you look hard enough. But is there a body of evidence that strongly supports an individual supplement for longevity for most people in the absence of some biomarker that you can measure? I'm not really convinced that there is at this point. I think you can make a reasonable case for NAD precursors. You can make a reasonable case for calcium alpha ketoglutarate, spermidine, you mentioned. Urolithin A, I think, looks pretty interesting. So I think there's a collection of these things. I think where part of the reason why so the other option would be just take everything, right? Part of the reason why I don't do that is I think that we don't have an understanding of the consequences of hitting this network in a bunch of different ways all at the same time. We don't have combinatorial studies, even in laboratory animals, looking at the impact of taking 10 or 20 different things all at once. And it might all be great. It might be synergistic, but my understanding of biological complexity leads me to believe that when you start randomly just messing around with stuff in a biological system, you're more likely to make it worse than you are to make it better. So that's sort of my default position. I want to be pretty confident that whatever I'm doing is either going to make it better or that I have a way to measure whether it's making it better. And so that's why none of the others have yet reached my personal threshold where I'm going to start taking them on a regular basis. And I also sort of feel like, again, I feel like I've gotten my diet pretty well dialed in. I've gotten my exercise pretty well dialed in. My sleep is pretty good. My stress levels are pretty good. So I might be the kind of person who would benefit from these supplements. I think the important thing for most people to appreciate is none of these supplements are going to make up for a crappy lifestyle. None of them. Those are the levers you want to pull first. And the idea that you can take an NAD precursor and eat garbage and not exercise is just nonsense. It's not going to move the needle. And I think people need to really understand that. But then you could get into each individual supplement and talk through the pluses and minuses. And all the ones I talked about, I think, are interesting. And maybe I'm making a mistake by not taking them. I don't know. But that's kind of where I'm at right now. I think there's and taurine's an interesting one. So here's a good case example. I was actually an author on that paper. I didn't do a ton of work for it, but I was an author on the paper that was published in Science showing that it could increase lifespan in multiple different animal models when supplemented. Even though I'm an author on it, it's one study. So I definitely...
Well, and it's in animals.
And it's in laboratory animals. Yeah, there was some human correlative data, right? Spermidine's the same way. There's interesting correlative data in people on dietary polyamines and health outcomes. So spermidine is of a class of molecules called polyamines. So I have been in this field long enough to just know some things stand the test of time, many things don't, and I want to see things replicated before I have, even though I'm a co-author on that paper, I want to see things replicated before I start really strongly saying, yep, this can go into that class, of take it to the bank interventions that we're confident modulate the biology of aging in an effective way.
And we can measure taurine. Taurine is easy to measure in plasma, so we can see people who need it. I routinely prescribe taurine when when there are individuals who don't appear to be making it well, which, incidentally include vegans and vegetarians don't
Yes.
appear. So it's conditionally essential. And in some of us, it's an essential nutrient, and we can measure it. So we
and
can
I think that's really important. I think the cases where you can measure and you actually know what suboptimal or deficiency look like, absolutely supplement to optimal when you can, for sure.
So are you engaged in some sort of a time-restricted eating structure or fasting program?
No, not at all. Partly, I don't really. I've never really. I've never enjoyed fasting.
Right.
But so here's the way I think about fasting and time-restricted eating. So the idea that those are longevity interventions, and again, I'm going to go back to the laboratory studies, because that's the only place where we can really definitively say that something is a longevity intervention, unless we have hundreds of years of human data that we can point to, right? The evidence that those things in and of themselves are longevity interventions modulating the biology of aging in an effective way in the absence of caloric restriction is essentially zero. There's a little bit with intermittent fasting, but it is small and hasn't been reproducible largely. And what I mean by that is, if you take a mouse and you feed them every other day, but you allow them to make up for the calories they didn't eat, they don't have a lifespan extension or a significant improvement in health span. If you also calorically restrict them, you do see a lifespan extension and an improvement in health span. So the idea that when you eat, in the absence of how much you eat, is a potent way to tweak this longevity network, I think is largely lacking. Does that mean there's no benefits for fasting and time-restricted eating? Absolutely not. And I think for some people, there are pretty big benefits. The most obvious being, for some people, it's a really useful way to control how much you eat, right? So I think if that works, great. I do think time-restricted eating for some people, especially if you are struggling with your circadian rhythm, there's some reason to believe that if you line up when you eat with your circadian rhythm, you can actually reinforce that in a positive way, improve sleep quality. So I do think there are some use cases for fasting, time-restricted eating that can have benefits for many people. But I don't think people should go into those things believing that they are significantly slowing biological aging in and of themselves. So it's an individual, I think, case for each of those. Again, I could be wrong. Maybe intermittent fasting does slow aging in people, but going off of what we know in the laboratory animals, in the absence of caloric restriction, there's not much evidence to support that.
Certainly clinically, we can see people who engage in it appropriately reap some benefits, in the appropriate, when it's prescribed to an individual who's probably somewhere on the metabolic continuum, if they can do it. We also see disordered eating. So that is, or orthorexia. So there's a whole new conversation happening, that wasn't, that I think is a fallout. It's a little bit of a fallout of this era of, only one meal a day. And, some of the, and we end up seeing some of these patients in our practice.
I think that's a really important point where the social and psychological interaction with food, humans are funny. I mean, we're funny animals, right? And so we can't match that. We don't look at that in laboratory mice, but it's real, right? And so you see that a lot. I mean, I don't know if you remember this group called the Calorie Restriction Society, but this was back in the early 2000s, right? There were a group of people who were self-practicing caloric restriction. They were sort of disciples of Roy Walford's. And they had this discussion group, and they were very open about, everything they were doing. But if you perused that discussion group, you saw a lot of what, again, I'm not a psychiatrist or psychologist, but what to me looked like a lot of eating disorders, right? Because of this whole social behavioral connection that we have with food. I think that's something that we don't always appreciate when people recommend these dietary modifications, that you do have to be aware of the potential psychological consequences that go along with it. And to what you were saying about time-restricted eating and intermittent fasting and metabolic disease, I mean, I think you're right. I think certainly there are people, and maybe this is true in general, where you will see an improvement in insulin sensitivity, particularly if you're already perturbed from these interventions. Now, it's an interesting question, and maybe you have a better feel for this from your clinical practice. I have not seen the definitive data. It's an interesting question whether or not those improvements in insulin sensitivity are completely independent of total calories or not? Or is it the case that there is those people who are practicing intermittent fasting are also eating less? I don't know the answer to that. It certainly could be the case that there are effects on insulin sensitivity independent of total caloric consumption.
It would be interesting to tease that out, just like we see different dietary patterns have influence on metabolic markers, regardless of the caloric,
right,
with isocaloric
right.
Structures. What did I want to say? Yeah, so when I did a postdoc in a clinical laboratory, and we had some of the caloric restrictor data back in early 2000s.
Yeah.
And it was, it comes to mind just an interesting pattern of amino acids in this population. And I recall, really like hypothyroidism, so just lower thyroid numbers. I don't know that matched energy. My my mentor at the time and teacher, Dr. Richard Lord, a nutritional biochemist, really felt like this was that they were healthy. He was in a lot of contact with them and thought that these were appropriate numbers for that population with such significant caloric restriction. Whereas in another model, you would definitely diagnose them with hypothyroidism.
Right. Yeah. And I think if you look at, again, there are several published papers. So John Hollisey, Luigi Fontana did a series of studies on these individuals who are self-practicing caloric restriction. And then there have also been the NIH funded calorie trials, which are more controlled. I think on whole, that data certainly points to the sort of canonical core set of biomarkers that you're going to get almost at any primary care visit, all going in the right direction in people practicing caloric restriction, right? Lipids look great, glucose looks great, insulin looks great, blood pressures looks great. So yeah, I think based on those biomarkers, they are healthier. And I certainly believe that caloric restriction, if done properly from an overall nutritional perspective, probably can slow biological aging in people. I think the question is, are there going to be corresponding detriments that offset the quality of life or quantity of life benefits you might get from slower biological aging? So we touched on the psychological consequences. You're also looking at things like loss of lean mass, loss of bone density, right? Yes. That could lead to problems in your 70s, 80s that would offset the benefits, even if at a sort of more whole body level, you are aging more slowly. I think we just don't know. And there are very few people, it's a very self-selected group of people who can maintain that lifestyle, right? Yeah. So it's not a pragmatic approach. We may find out with the GLP-1 agonists, because now we're starting to get this not natural is the right word, this population of people who are being pharmacologically aided in caloric restriction. We may find out over the next couple of decades if there are negative consequences that more than offset the health benefits from these drugs. I don't think they will more than offset in an obese population, but if we have a bunch of normal weight people start taking these drugs, we may find out. Yeah, right. Well, much remains to be unfolded. Yeah. Listen, we could keep talking. I appreciate your insight into just a really broad range of topics today. Thanks for joining me, Matt. You're welcome. My pleasure. As always, thank you for listening to New Frontiers in Functional Medicine, where our sponsors help bring the very best minds in functional medicine. And of course, today is no exception. Not everybody can be a sponsor on my platform. I appreciate the good work, the relentless research, and the generous support from my friends at Rupa Health, Biotics, Integrative Therapeutics, Timeline, and OneSkin. These are brands I know and trust and use in my clinic and can confidently recommend to you. Please visit them online and let them know you learned about them on New Frontiers. And if it's not too much to ask, consider leaving a thumbs up and a kind review wherever you're listening to New Frontiers. Thanks.